Good afternoon, everyone. My name is Katie Kerr. I'm a postdoc at Queen's University Belfast, and it's a pleasure to be here today at the Irish Society of Human Genetics. And I'm going to be talking a little bit about Northern Ireland's participation in the 100,000 Genes Project and some of the lessons we've learned about whole genome sequencing from this. So, many of you will have listened to the talk this morning from Professor Dane Hill about the 100,000 Genes Project, but just for anyone who wasn't at the talk, 100,000 Genes Project sought to perform whole genome sequencing for about 100,000 individuals, and this comprised individuals with rare diseases, their family members, rare cancers, and some infectious diseases. In 2015, a collaborative effort between patient-led organisations such as the Northern Ireland Rare Disease Partnership, clinicians and researchers allowed Northern Ireland to join this project. And we recruited about 1,000 participants, including about 440 rare disease probands. I'm only going to be talking about the rare disease element of the project today, not about rare cancer. The project recruitment and the initial analysis completed in 2020, somewhat later than the 2017 that we had initially projected, but it was a pilot study and this was to be expected. And we had a diagnostic yield of approximately 22%. As part of this project, we conducted a translational research workstream with the aim of evaluating the general outcomes from Northern Ireland's participation. This slide just gives you a workflow of how the 100,000 Genes Project worked in Northern Ireland. So participants with suspected undiagnosed rare or genetic diseases were recruited through a variety of medical specialities as we wanted this to be a multidisciplinary genomic service. Blood samples after recruitment and consent were extracted and outsourced to Genomics England for whole genome sequencing. In addition, we collected approximately 800 omic samples for later functional analysis where appropriate, and I'm going to be talking about that a bit later on. We then had the sequencing results returned and bioinformatically stratified into 3 tiers. Again, I'm going to be talking about this in more detail later on. And the variant investigation was conducted by clinical scientists in a multidisciplinary fashion within the Northern Ireland Genomic Medicine Centre, and validated results were reported to the probands and their family members by clinicians. Some of the key discussion points that I'm going to be talking about today include the comparison of the NIGMC outcomes to the rest of the UK, how we can improve diagnostic yield, and how we can better facilitate the integration of genomic medicine into multidisciplinary healthcare moving forward. So the diagnostic yields and the operational challenges that we faced in Northern Ireland were comparable to England, Scotland and Wales. In particular, you can see our diagnostic yield of about 22% was very similar to Scotland, England and Wales, which was good to see. Some of the operational challenges that we faced included a limited workplace capacity. Our clinical scientists were only able to process the top 2 tiered variants per proband, with a limited capacity to delve into lower tiered or untiered variants. And these could still have biological relevance to the patient's phenotype, and again, I'll be talking about that in a bit more detail later on. We also found the research collaboration potential was not maximised, and that should have been one of the key advantages of setting up the Northern Ireland Genomic Medicine Centre, was facilitation of collaborative academic and clinical research through the Genomics England clinical interpretation partnerships, but this never really got off the ground the way we had envisaged. And to this end, we're conducting a project to explore what the barriers were for people in participating in these projects and what we can do to facilitate future collaborative genomics service development research moving forward. How can we improve diagnostic yield then? As I say, we're currently sitting at approximately 22%, and one of the key advantages of whole genome sequencing data compared to traditional microarray diagnostic tests is that once you've collected the data, you have it, and it can be reanalyzed as your knowledge improves over time. And this international standard for reanalysis of whole genome sequencing data is about every 18 months. Having said that, we may still be able to tease out a few further diagnoses from the data and the biological knowledge we have now. And 2 of the ways that we can do this are through investigation of lower tiered and exomizer variants, or through conducting additional multi-omic analysis to provide further functional evidence. So this would be, for example, whenever you have a variant of unknown significance that you think might be related to the patient's phenotype, but you just need a wee bit more biological plausibility to push it into that pathogenic criteria. So this just gives you an overview of what the tiering process was by Genomics England, so we're all on the same page. The raw sequencing data was generated by Genomics England and bioinformatically filtered into panels that contain genes associated with diseases. Tier 1 and 2 were the most likely to be associated with the disease, and therefore these were the ones that were focused on by clinical scientists within the NIGMC. Whereas Tier 3 contain variants of unknown consequence, but they could still have relative or relevant phenotypic associations. Variants were additionally ranked by Xmiser, which is a software that ranks variants of unknown significance by prior phenotypic associations, and previous research has shown that you can in fact sometimes find a diagnosis from looking at just Xmiser variants alone. So in the NIGMC, we have within the 100,000 Genomes project approximately 350 probands that remain undiagnosed. And so a workflow is currently in development for the follow-up of these undiagnosed participants in a collaborative manner by clinicians and researchers, including looking at the top 5 Xmiser variants and conducting further omic analysis. Just to give you an example of how powerful Xmiser can be in identifying the pathogenic variant, this depicts a proband that had structural abnormalities, including agenesis of the corpus callosum. The participant had no Tier 1 or 2 diagnosis, but they did have a compound heterozygous variant within the gene ROBO1 that was ranked 1 by the Xmiser software. And this paper here shows that there was a strong prior association with the gene ROBO1 in the patient's phenotype, and therefore this was likely to be pathogenic, even though it didn't come back through gel tiering. This depicts an example of the power of omics in providing a diagnosis. So a family with the condition hereditary hemorrhagic telangiectasia was recruited to the project, and the research was conducted collaboratively with the respiratory GSIP led by Professor Shovlin. The proband had serious pulmonary malformations and had previously tested negative for variants within known ISHG genes. There was no Tier 1 or 2 variants, but there was 76 Tier 3 variants. This included a variant within the gene GDF2 that encodes the protein BMP9, which is a ligand for a well-known HHT gene ALK1. Additional proteomic analysis with ELISA found that the BMP9 levels did not form the mature protein as a result of the GDF2 variant, and the levels were significantly lower in the affected family and particularly low in the affected proband, and therefore this GDF2 variant was identified through additional omic analysis of lower tier variants to be a rare variant associated with the HHT phenotype. How can we then better facilitate the integration of genomic medicine into multidisciplinary healthcare moving forward? We identified that probands which were recruited to the genetics clinic were almost twice as likely to receive a diagnosis compared to all other medical specialities combined. There are several potential reasons for this, perhaps including the resources that are already available to geneticists, such as prior established collaborative networks, and for example, the Deciphering Developmental Disorders study, which was previously conducted within the genetics clinic. Anecdotal clinical genetics expertise on selection of variants for follow-up, perhaps variants that they've encountered in the past. And then the bit that we were interested in the most was a variation in phenotyping depth, which can impact diagnostic yield. And actually we discovered that there was a huge significant difference in phenotyping depth between genetics clinic, with approximately 10 HBO or human phenotype ontology terms reported per participant in the genetics clinic compared to approximately 4 HBO terms reported per participant in all other clinics. And this really emphasizes the importance of deep phenotyping to improve diagnostic yield from whole genome sequencing data, and that we need to develop our expertise beyond the genetics clinics alone. Just to conclude then, the top lessons from the 100,000 Genomes project in Northern Ireland. There's significant promise in whole genome sequencing for the future, but there's a lot of work to be done before we can allow it to be integrated into mainstream healthcare. There's a need and opportunity to conduct collaborative translational academic clinical research, and we've shown and proved that this can actually aid diagnosis of probands and facilitate carrier testing through omic analysis or analysis of Xmiser variants. And finally, we need to prioritize genomics education right through from undergraduate level to postgraduate level, but also across all medical specialities if we want to provide a multidisciplinary genomics healthcare service. Thank you very much for listening, and a massive thank you to all the participants of the 100,000 Genomes Project and to the Northern Ireland Genomic Medicine Centre for allowing me to conduct this translational research, and I would be glad to answer any of your questions.